Alright, I'm going to start with the light reactions, and it's called the light reactions because it's light dependent, which means it needs a photon of light to really get it going. That's the energy source. Okay, so right here I started with H2O plus CO2 plus light because basically what all you need for photosynthesis to occur. And then CO2 goes in the plant, and oxygen, which is O2, leaves, and it comes out of the stomata. And down here, a photon of light is absorbed through chlorophyll, and chlorophyll is stored in, where is the chloroplast? Well, I guess I didn't write chloroplast, but it's stored in the chloroplast, and chlorophyll is really in the thylakoid, which is in the chloroplast, and the thylakoid has a thylakoid membrane and a thylakoid space, and the space that's like outside of the thylakoids, well, a stack of thylakoid is like a grana, so outside of each like little grana is like a aqueous liquid, and it's called the stroma. So back to photon. Whenever a photon of light excuse me, whenever a photon of light hits chlorophyll, it basically gets kind of excited, and it excites the electrons, and then it causes the electrons to kind of like leave the spot it's in. So here, the photon of light excites, well, it goes through the chlorophyll and it excites the electrons, and then the electrons leave the chlorophyll and hop onto a protein, which causes the, needs to go to H2O. It causes the H2O to split, which is like a really hard thing to do, which is why it's quite amazing that it actually does this like on a daily basis. But it splits into oxygen and hydrogen ions, and basically whenever it does that, that's part of that's like the beginning of photosystem 2. It starts with photosystem 2 for some weird reason. And from photosystem 2, basically that's what happens. It's just like the splitting of the H2O into the oxygen, which is like the byproduct, and the hydrogen ion. It goes through the cytochrome B6F complex, and basically it's like a intermediary complex between the photosystem 2 and photosystem 1. And also it uses energy from like the electrons that got excited and like left on the protein. It uses those electrons to pump proteins into the thylakoid space because it's all in the thylakoid like area, but it pumps it into the thylakoid actually. Alright, so this is the cytochrome B photosystem. Okay, so right here we have the photosystem 1, and protons are, so the thylakoid, okay, yeah, because it pumps electrons to the thylakoid. Thylakoid is over here. Thylakoid is like, it gets like charged up because they put so many like protons and stuff into the thylakoid, so it kind of pumps it and like charges it like a battery, and then when it's full of protons, it creates a concentration gradient, and then the protons like basically push away from each other to try to get away, and it makes their way through an enzyme on the thylakoid membrane, and that makes its way through ATP synthase. And ATP synthase is basically like when a phosphate attaches to the ADP, it creates ATP. And in photosystem 1, again, the electrons get re-energized, and photons like excite the electrons again, and then they leave again, and then all energy is used to make all energy basically that is re-energized from like photosystem 1. It helps to make NADPH, and that's basically made from NAD plus and two electrons and a hydrogen ion, and oxygen is always the byproduct. So that's my little flow chart for light reactions. So I'll flip this over and I can go over the Calvin cycle. Okay, so here I have the Calvin cycle, and it's like nicknamed as like the dark reactions, which is kind of ironic because it occurs during the day. It's not like at nighttime, which a lot of people probably think if they don't really know. But it doesn't require a photon of light, so it's independent. So like independent actually would be the best description of it, rather than like dark reactions, because it's kind of misleading if you're not really paying attention. Alright, and energy here, since it doesn't come from light, it comes from the NADPH and the ATP that we created like in the light cycle. And I just gave a little refresher. The ATP is made from the ADP is joined with like a phosphate, and then the NADP plus is at like it's added to 2 electrons and a hydrogen ion to make the NADPH. So the Calvin cycle really begins in the stroma, and the stroma is in the chloroplast, and that's where carbon fixation begins. Oh, in the chloroplast, you know, inside the chloroplast is the thylakoid and the chlorophyll, thylakoid membrane, and all that other good stuff. But it mainly just kind of sits in the chloroplast. It doesn't really say specifically where, but in the stroma, that's where the carbon fixation begins, and basically fix the CO2 with RUBP, which is ribulose bisphosphate. And the ribulose bisphosphate is always in the chloroplast because that's like kind of like the start and the finish of like photosynthesis. All right, so CO2 is fixated with ribisco, which is an enzyme that converts inorganic carbon into organic carbon. And so whenever the CO2 is fixated with the ribisco, which right there, it causes it unstable. It makes it like unstable, so it turns it into the 3-phosphoglycerate. And that is really like the first stable molecule in the Calvin cycle. So then in the second phase, which is called reduction, I have reduction right here, we use the energy, we need some more energy basically, because we use up all the energy that we started off with. And since we don't have a photon of light, we have to like get more ATP and NADH. So the ATP gives a phosphate group to the 3-phosphoglycerate, which was created after we fixated the carbon and the ribisco together. So it gives, the ATP gives a phosphate group to the 3-phosphoglycerate, and then the NADH gives electrons, and then what happens next? Oh, it gives it 2 molecules of the glyceraldehyde 3-phosphate, which is also nicknamed G3P, and that's basically a high energy 3-carbon compound that plants can use to convert like carbohydrates into basically anything. So here I have carbohydrates, and carbohydrates are glucose, cellulose, and starch. Those are just some examples. And basically in a plant, the carbohydrate can be converted into glucose, and glucose in a plant basically functions as like short-term energy storage. Cellulose is for like help with the structure, and starch is like long-term storage. And then the glyceraldehyde 3-phosphate is the ultimate product of photosynthesis. That's basically what we're trying to get out of it. But in order to like get it, we need like 5 G3Ps. We need 5 G3Ps to regenerate the 3 RUBPs, which is the ribulose bisphosphate, that we started with. We need 9 molecules of the ATP and 6 NADPHs. So in order to generate the 3 RUBPs, and so we have to get all that in order to generate the 3 RUBPs into 6 G3Ps. And the G3Ps are the glyceraldehyde 3-phosphate. But only really one can leave because the rest are needed to like restore the like energy that we already used, because we have to like kind of replace it. It's just like a give and take. Like we borrow the energy, we got to give it back. And basically after that we go to the last phase, which is regeneration, and that's basically how plants use the water and the light energy and the CO2 to turn it into everything that is living that we use today. So those are my 2 videos in one.